Hi everyone, welcome to lecture 7. This lecture's topic is paleoecology. This is the second and hopefully final recorded lecture. And paleoecology, what is it? We'll talk about all that, but this is a topic that's a bit near and dear to my heart because I don't know if you remember from the very first lecture I gave in this class, I mentioned that paleoecology is my particular specialty within paleoanthropologists. And one label I use to describe myself is paleoecologist. So a good portion of this lecture topic is what I study for my research. Okay, and the lecture outline for today. So what is paleoecology? We'll define it. What is its role in paleoanthropology, which of course is the topic of this class, so that's very important. And paleoecology can be roughly broken up into two categories. One is paleoclimate. Okay, we'll talk about how that's reconstructed. And we'll talk about some famous paleoclimate hypotheses in human evolution, which you may or may not have been exposed to. And paleoecology being broken down into one, paleoclimate, and two, reconstructing paleoenvironments. I'll talk about those categories and kind of where that distinction is. Okay, so what is paleoecology and what is its role in paleoanthropology? So paleoecology, we could break down this word, right? The prefix paleo, you've encountered before, we've defined before. Okay, it comes from Greek, the word paleos, which means ancient. So particularly in the context of this class, but really any biological class, paleo usually refers to the fossil record, okay? And in the context of this class, it's going to be hominin paleoecology, but paleoecology can be applied to any fossil taxon. It could be dinosaurs, trilobites, ancient clams, you name it. Okay, the suffix ecology, what does that mean? So there are kind of two definitions I see commonly used to define ecology in ecology textbooks. One, and the one we're really going to be focusing on, is presented here. The second one I'll bring up briefly in a bit, but the primary one, yeah, the study of the abundance and spatial temporal distribution of organisms and the factors influencing these patterns, okay? So abundance, why is a particular species so abundant in one site but rare in another? Okay, spatial temporal distribution, why are species found in certain parts of the earth and at certain time periods, years maybe, but not in other parts of the earth or other years, okay? So that's really what we're talking about here and the factors influencing these patterns. Okay, so for example, here is a map of Africa with these different species of baboon. Okay, you see they occupy certain parts. So this is Papio ursinus occupying South Africa. Why is Papio ursinus here, but not here in Northern Africa? Okay, that is in the realm of ecology. Why is that? What factors are influencing this spatial distribution? And abundance, this is, if you ever study ecology, particularly community ecology, this is a popular figure. This is known as a species abundance distribution. Okay, on the y-axis, we have number of species. On the x-axis, abundance. Okay, what we see here is, and this is really a general phenomenon across all areas of the globe, across all time periods, even extending into fossil record time periods, across all taxonomic groups. What we see is that most species are rare. Okay, so towards the left on the x-axis is low abundance. We see most species fit in this category of low abundance, few individuals, and very few species, you see these bars are short, have large abundances towards the right on the x-axis. Again, very general phenomenon. Basically, most species are rare, very few species are common. Why that is, still a big puzzle in ecology. So when you apply this definition of paleoecology to hominins, you get hominin paleoecology. And so adapting that definition previously, hominin paleoecology is defined as the study of the abundance and spatial temporal distribution of fossil hominins. Okay, and the factors influencing these patterns. So for example, why are hominins found very commonly in Africa, and particularly north, east, south, okay, but maybe not here? And why are they not so common in, for example, South America? Obviously, based on the previous lecture, some of this is due to taphonomic processes, but some of it is due to a quote unquote real ecological signal. Right, and hominins, this is a hominin family tree, if you will, right? Time is on the vertical axis. And so why are there so many species of hominin in this time period, right? We have the genus Paranthropus, we have the genus Homo. Okay, why do they appear at this time period? Why are they not found earlier? Why is Paranthropus not living today? Okay, these are all questions in the realm of hominin paleoecology. So how is hominin paleoecology traditionally done? Okay, for the most part, hominin paleoecology equals paleo-environmental reconstruction. So in the field of paleoanthropology, paleoecology is synonymous with reconstructing these ancient environments that these hominins lived in. Okay, and what this involves is going to a fossil site. So this is one I've mentioned previously, Olduvai Gorge, located in Tanzania. Okay, you go to the site, you scour these outcrops, and you might find certain fossils. Okay, this is a ancient zebra relative. This is fossil pollen. Okay, this is something called soil carbonate, which I will expand upon and talk about more later. And from these fossils that you find, you then use that in pretty creative ways to reconstruct these ancient environments that hominins lived in. Okay, so that's what I mean by paleoenvironmental reconstruction. You reconstruct these ancient habitats, aka environments that these hominins lived in, using the fossils that you find at fossil sites today. Okay, so why the emphasis on paleoenvironmental reconstruction in paleoanthropology? In some ways, this makes a lot of sense, and you could probably already guess. But right, from Darwin, we know that the environment places selective pressures on organisms. Okay, the classic example is Darwin's finches, right? Why do Darwin's finches, their beaks look the way they do? Darwin connected that to the different types of habitats these finch species occupied and the selective pressures imparted on their beak morphologies, right? And you've probably encountered this multiple times by now, but right, those finches that live in trees and eat insects, they have beaks shapes that are better for grasping those insects. Those that live and feed on, let's say, the nectar of plants or cactus, right? Their beaks are a little bit more slender for kind of probing, getting into the cactus, eating the flesh and the water. And those finches that live in habitats where they exploit things like hard seeds, they have more robust, kind of deeper and shorter beaks, right, to crack open those seeds. And so these different food types in these different environments, right, will select for certain beak morphologies. And these are the selective pressures imparted on these finches by the environment. So then making this connection with hominins, presumably then reconstructing paleo environments may shed light on the selective pressures faced by ancient hominins and may give us an idea. We could begin to hypothesize or even test hypotheses for why hominins look the way they do, have a certain morphology. Perhaps it's due to these selective pressures due to the paleo environments, these ancient environments that these hominins lived in. Okay, so paleoenvironmental reconstruction. You can kind of broadly dichotomize this into two scales, okay, two spatial scales. One is very large scale, okay, regional, let's say the continent of Africa or all of East Africa to global, okay. We're going to call this paleoclimate, okay. So this is the kind of dichotomy I was referring to in the outline at the beginning of the lecture. And you can kind of connect it to today, right? We often talk about like whether being the day-to-day change in temperature and precipitation, for example. Climate is the kind of longer term averages and trends that we see over many years, many decades, right? So you could kind of draw a parallel here. That large scale is reflected here, although spatially, we're going to call that paleoclimate, reconstructing these environmental variables at these very large spatial scales. Okay, paleoclimate you could define really broadly as ancient climate patterns and their change through time across space. And we have the smaller scale, okay, smaller than a regional, let's say smaller than the region of East Africa. Typically, we use the words local, this idea of a depositional basin. If you don't know what that is, that's okay. Let's just talk about a site, okay? So when you're reconstructing these environmental variables at this smaller scale, we can say that's a paleoenvironment, which can be defined as ancient environmental patterns and change, okay? So just like with paleoclimate, this is the definition we're going to use. Okay. And so there are many ways, and we will go over these different ways that people have reconstructed global scale, in this case, temperature change through time, okay, from over 4 million years ago to the present, okay, at the continental scale, in this case, either Africa, or in this case is more specifically East Africa, right? We also have a reconstructed precipitation / aridity curve through time. Okay, we'll talk about how that's done. This one, basically seeing which sites or basins at which time periods have lakes. We won't really go over that. And yeah, at that more kind of local scale, this case is that basin. Yeah, you could think of it as just a really large site. This reconstruction of vegetation change through time, going from more wooded to more of an open grassland. Okay, so yeah, just I guess to emphasize here that this idea of reconstructing these environmental variables are done at very different scales, going from the broadest you can totally go, global, to the continent of Africa or the region of Eastern Africa, all the way down to the site scale. Okay, and I kind of made this distinction maybe around here-ish. Okay, paleoclimate, paleoenvironment, but basically trying to do the same thing, asking the same questions, just at different spatial scales. Okay, so remember how I said there are two very common textbook definitions of ecology. I gave one previously, which is the one we're really going to use. But here's another common definition of ecology. Maybe you've encountered this. Ecology defined as the study of the relationships among living organisms and their physical environments. Okay, so studying how organisms interact with each other and how these organisms interact with their physical environment. Okay, these relationships and interactions among living organisms, for example, include things like inter or intra-specific competition for resources, right? So if a zebra is competing with a wildebeest for grass, that's inter-specific competition, different species, or if it's a herd of wildebeest competing with each other for grass, right? Competing within a species, but different individuals. These also include predator-prey interactions, right? Like a lion eating a zebra. And in the realm of paleoanthropology, these interactions, competition for resources, predator-prey interactions, they're typically not in the realm of what we traditionally consider paleoecology within paleoanthropology. Interestingly enough, probably for historical reasons, the study of these interactions is relegated to the field of zooarchaeology, okay, which is that subdiscipline of archaeology that focuses on the analysis of animal remains from archaeological contexts. And so you may or may not have taken a class on this. I believe Michael Ponte or one of his PhD students teaches one. Faunal analysis is the one I was thinking of. But it's because what they do is when they go into the fossil record, right, they look through basically kind of like a microscope at the surfaces of the fossils, and they're looking for these marks. And so this is a trace fossil, right? Although technically this one is an experiment. So this is done on what was once a living animal. but definitely not an extinct, long extinct organism. But here we see tooth marks, right? And so if this bone eventually becomes fossilized and discovered, it becomes a fossil, right? We could still see those tooth marks on the fossil surface. And again, this is a trace fossil, and it's a preserved direct evidence for predator-prey interaction. Okay, some carnivore with its tooth was taking meat off the bone and accidentally scored the bone with its tooth, leaving these marks. Okay, basically predator-prey interaction. Here's another one, a cut mark, right? So in this case, a hominin with a stone tool, cutting meat off the bone, accidentally scoring the bone, leaving a cut mark. Another example of predator-prey interactions. Okay, and percussion mark, same thing. Hominin using a round stone to break open a bone to get at the fatty marrow within long bones such as the femur or the humerus. Okay, if you don't know what those terms are, don't worry, you'll learn that soon. But again, another evidence of predator-prey interactions. And so zooarchaeologists within paleoanthropology study these what are known as bone surface modifications to get at the study of these predator-prey interactions, but also to get at how hominins, for example, competed with other carnivores for mammal prey. And this is the whole like where these early hominins, when they first ate meat, were they hunters, were they scavengers, right? That implies or let's just say competition for these mammal resources, their flesh, their bone marrow. And again, all this is typically related to the field of zooarchaeology. So I won't talk about bone surface modifications or interactions among organisms anymore, but there is a class that we offer in our department that covers this stuff. And there is a researcher who studies this stuff, and that's, yeah, Michael Ponte. Okay, so let's go over paleoclimate. Again, that really large spatial scale study of these ancient environmental / climatic variables. So reconstructing paleoclimate, okay, one of the earliest and probably still most common ways of doing so is using something called a deep sea core, okay? Basically, I think I have a picture which I show on this slide. You have a ship that goes out and it sinks a massive, basically kind of metal cylinder into the ocean floor, okay, which comprises sediment that has basically rained down and is slowly deposited over time. So by sinking that core into the seafloor and extracting all those different layers of sediment, aka strata, you're basically getting layers through time. And then we could study that. Specifically, what we study is a ratio of an oxygen isotope, 18, which means it has, oh God, I forget this, atomic mass of 18, I believe. So it has a total of 18 protons and neutrons, okay? Ratio of that heavier isotope of oxygen relative to this lighter isotope, 16, which is the most common one. I believe that's 8 protons and 8 neutrons. And again, an isotope is just an atom, these elements, in this case oxygen, which have the same number of protons, these two, just different number of neutrons and therefore different weights. And this ratio is basically summarized in this notation. This is a Greek delta, delta O18. Okay, so that's very common in stable isotope analyses. You'll see that letter, delta. If you really want to be fancy, people abbreviate this as DeLO18. But it's basically the ratio of the heavier oxygen isotope to the lighter one. And what you can see here, right, we have through time, from a long time ago, okay, into, I mean, basically past 65 million years ago. So dinosaurs are still alive here, all the way to the present. And what we see is this measure of DeLO18. The unit is something called a parts per mil. Okay, so you kind of recognize a variant of this, the percent symbol, which is 1, 0, and then a slash, and then 1, 0 in the denominator. Okay, percent per 100. Add another 0 in the denominator, it becomes per mil, per 1,000. So that's really what that is. Okay, parts per 1,000 parts at a time. And What you see is this, yeah, Delo 18 measured in parts per mil, how it changes through time. Okay, but what this is a measure of temperature. How does this work? Well, as temperature increases, and we're talking about global scale, right? So as the global scale temperature increases, okay, the lighter oxygen 16, because it's lighter, gets preferentially evaporated off, thus leaving more oxygen 18 relative to oxygen 16. Thus, this ratio increases, thus this measure increases. Okay, so we could see here, as this increases, the temperature should increase. Oh my God, what is going on here? I think this should be more positive. I wonder if these are supposed to be negative or something like that. Anyway, yeah, I'm going to look into this, but yeah, I'm almost certain as you go towards the right, this is increasing, which is associated with a higher temperature. Okay, and what you do is in these deep sea sediment cores that I mentioned, in each layer, you have these microscopic organisms called foraminifera, and benthic just means like the depths of the ocean, like barely above the ocean floor. And forams are these microscopic organisms that build shells out of the minerals found in the surrounding seawater. And those minerals have oxygen isotopes, okay? So they're basically recording the oxygen isotopic composition of the surrounding deep ocean water, okay? So they're recording that oxygen isotopic composition of deep water for that time period, okay? And then they die, still recorded in their shells. I mean, these guys are microscopic. You probably can't even see it, but each of these scale bars is a micron, which is 1/1,000,000th of a meter. Okay, so really, really, really small. But what you do is you can analyze them chemically, a bunch of them through time, right? Because again, those deep sea cores have many layers, and then that's how you could reconstruct this ancient temperature curve. Okay, yeah, okay, so here's an example. Yeah, you have these ships that go out into the middle ocean. They sink these big metal cylinders, and what they get, they sink these these empty metal cylinders into the ocean floor. They pull it back up, and inside the empty cylinder, what was once empty is now filled with the sediment, different layers representing different time periods. Okay. And you split it right down the middle, and you take samples at every so often, look for forams, chemically analyze them, estimate and calculate their Delo 18, and you can construct this temperature curve through time. Okay, here's another example, dust cores, basically using those deep sea cores again. But how this works is, well, you use those deep sea cores and then you look at these different time periods and see the percentage of dust. It's really that simple. How does this work, or why are we interested in the dust? Well, when the climate becomes more arid, vegetation cover decreases. Okay, and whether you know this or not, one function vegetation serves is its roots kind of anchor soils down. So when you have vegetation, you don't get soil erosion as much because those roots are holding that soil down and in place. But when the climate becomes more arid and vegetation starts to die off, those roots go with it. Now that soil, especially at the top soil is loose and can be blown away, and it forms dust, okay? And that dust gets blown into the ocean. So here, it's not a great figure, but this is a map of Africa, okay, Asia, and you can see where those ships have made journeys and sunk cores in. Okay, so we have cores here off the coast of West Africa, some off of East Africa. Okay, so like for example, these cores are particularly recording the dust blowing off of Western Africa. So this is recording not so much global, but more of a regional signal. Okay, in this case, probably stuff that's just coming off of West Africa. this Eastern Africa. Okay, again, you sink those deep sea cores, you split it down the middle, and then you sample different time periods of the sediment, and you could calculate the percentage of dust in these different time periods. And then you can calculate curves like this. So through time, right, 4.5 million years ago to the present, you can see each one of these sites equals a different core. So 659 is there, 661 is there, and you can see how the percentage of dust changes through time, again, reflecting that arid climate. So more towards the right is more dust, and thus more arid. Towards the left, the opposite. So that's how that works. Okay, there are many things that control paleoclimate, just like there are many things that control climate today. But one popular or strong control that is often taught in classes like this is something called the Milankovitch cycle. Okay. So Milankovitch cycles, there are 3 of them, and they operate at different time scales. So one is called precession. So whether you know or not, right, the Earth rotates on an axis, and that axis actually wobbles around like this, like a spinning top. Okay, and it does so on time scales of about 20, I think it was like 23,000 years old, 23,000 years. So one full rotation takes about 23,000 years. Okay, that's one type of Milankovitch cycle. Another one is something called obliquity. So that axis upon which the Earth rotates actually swings back and forth like this. Okay, one full cycle happens on the time scale of 41,000 years. And then eccentricity is the shape of the Earth's orbit around the sun, from circular, okay, all the way to very elliptical. And then back, one cycle takes 100,000 years. Okay, so there are these 3 different types of Milankovitch cycles that happen at 3 different time scales. And what it does is effectively, as you can imagine, when you change each of these 3 different things, okay, you're changing the amount of solar radiation received on the planet at a given place at a given time, okay, which then in turn influences atmospheric circulation, temperature, basically these climatic variables, right? And so Milankovitch cycles have been shown to influence paleoclimate. Like the curve I showed, what was it, 2 slides ago, that Dello 18 through time, right? There's a lot of squiggles. I mean, even the dust curve, if you can statistically decompose it, you can see that there will be a signal that is 23,000 years, 41,000 years, 100,000 years. Okay, so this is actually a strong control on paleoclimate. When you're at higher latitudes, maybe you can already kind of intuit and see this. When you change this axis of tilt, going from 24.5 degrees to 22.1 degrees, okay, that mainly influences the amount of solar radiation received towards the poles. Okay, what's more important for near the equator is precession and eccentricity, and particularly early hominin evolution. Okay, closer to the equator, precession and eccentricity is going to be more important. Okay, so some famous climate hypotheses in human evolution. Don't know why this box is hanging out here, but first, some quick background. Okay, this is that Dello 18 curve through time. Okay, you can see there is a trend. It goes from towards the right to the left, and the squiggles, the back and forths, right? Again, driven by these Milankovitch cycles, becomes more pronounced through time. Okay, so the takeaway here is that climate becomes more arid and variable over the time periods that cover human evolution. Okay, and you could see at which time periods the variability cycle is following. Right, this is going to be more. Precession is more important from like 3 million years and earlier. From this time period, it's going to be obliquity. Although again, that signal is a lot more attenuated near the equator, which is where a lot of early hominin evolution takes place, and later the eccentricity cycle. And yeah, this paper is explicitly connecting all of this to human evolution. Okay, so that's just some background, right? Basically, I just want you to take away from this is that climate becomes more arid and variable through time. Okay, so the first famous savanna hypothesis was put forth by Charles Darwin, okay, in excuse me, Descent of Man, published in 1871. This is known as the savanna hypothesis. And so the savanna hypothesis basically says that with climate change, you get closed, more treed forests, so closed canopy, and that slowly gives way into an more open savanna grassland environment, something that looks like this. And it's hypothesized that such a dramatic change in the environment presented new adaptive challenges for hominids. And the big one that was posited was bipedalism, move around and walking on two legs. And so it's thought that bipedalism is more efficient for moving around in more open environments like grasslands. And we'll cover why that might be the case when we actually go over bipedalism in a future lecture. So yeah, put forth first by Darwin, but also really made famous by Raymond Dart, who maybe you know that name. He was the one that discovered Australopithecus africanus, or named it, and was the one who discovered the Tong child, or someone else discovered it and handed it to him. I'll tell you that story when we get there. But yeah, this is actually his paper naming Australopithecus africanus, published in 1925. And I want to read this quote near the end of the paper. For the production of man, a different apprenticeship was needed to sharpen the wits and quicken the higher manifestations of intellect. A more open veldt country, savanna, where competition was keener between swiftness and stealth, and where adroitness of thinking and movement played a preponderating role, I don't think that's a word, in the preservation of the species. Darwin has said, no country in the world abounds in a greater degree with dangerous beasts than Southern Africa. And in my opinion, Southern Africa, by providing a vast open country with occasional wooded belts and a relative scarcity of water, together with a fierce and bitter mammalian competition, furnished a laboratory such as was essential to this penultimate phase of human evolution. Okay, so here Darwin, in very floral language, Dart, make a connection between the opening up of these environments, going from forests to savanna grasslands, produce these new adaptive challenges that really selected for these key adaptations that defines hominins. Okay, another very popular climate hypothesis is something called the turnover pulse hypothesis. It was put forth by Elizabeth Verba, and Elizabeth Verba was really influenced at this time period. We were starting to get some pretty good paleoclimate data, okay, and then so we could see these time periods when you get these massive shifts towards these cooler, more arid climates. And what Verba noticed, because Verba's specialty was studying antelopes, was that there were these time periods when you have this cluster of extinctions, particularly of these non-grassland species of antelopes, and you get the appearance and speciation of the grassland-loving antelope species. Okay, this temporal cluster of extinction speciations, this is known as a turnover pulse. So here's a figure from one of her papers, okay? And you could kind of even see it yourself, right? So this is time on the vertical axis. On the x-axis is just the different lineages of these different antelope species. Okay, here we see some terminate. Here we see some speciate, or I should be more accurate. This is when they disappear from the fossil record. This is when they appear in the fossil record. And probably by eye, you can see that there's kind of a cluster here, a line you could draw, okay? 2.5 million years ago. Would you know it, that's the time period when you start getting the onset and intensification of glaciers forming in the Arctic in the northern hemisphere, okay, which really influence paleoclimate. And so Verba made a distinct connection that global climate change at this time period basically caused the extinction of these non-grassland loving species and the speciation of these grassland loving species. And she did connect it to human evolution. very explicitly in a paper that was published 3 years later. Okay, in fact, she connected this exact pulse to causing the appearance of the genus Homo, okay, due to this global climate change pulse towards cooler, more arid climates. Okay, this is the third and last hypothesis I'll cover. This is something called the variability selection hypothesis as put forth by Rick Potts. Okay, what Rick proposed, and again, he based this off of what he noticed at his own study area, this site called Olorgesailie Island in southern Kenya, that looking at the geology, you can see climate take these drastic swings when climate is very variable, and these other time periods when climate is quite stable. And Rick, who also studies the mammal fossil record, noticed that there were certain extinctions and speciations that were associated with these times of high climate variability or stability. So this is very different from that savanna hypothesis and turnover pulse, which emphasizes directional change of climate, right? Going from warmer, wetter, sharp change to cooler, arid, and grasslands, right? Rick said, no, we shouldn't be focusing on these trends. In fact, we should be focusing on the variability, the amount of variation. And what he saw and hypothesized is that during times of high climate variability, what you get is extinction of specialists and the speciation of generalists, okay? Because if climate is swinging from moist or warm and wet to cool and arid, and that's happening very quickly on small time scales, then he says those animals that are really specialized for one of those climate conditions, they just can't keep up with that much change in that short of a time period. And what you get instead is the speciation and survival of generalists, those that can survive and do well in either kind of climate mode. Okay, and so yeah, this is a figure from one of his papers. So again, this is that del O18. So this is del O18 curve going from older to the right, younger to the left. And so again, we see these swings become much more, the amplitude becomes higher through time. And he notes in purple these time periods of high climate variability. And he connects it to things like, yeah, the speciation of certain hominins, the extinction of certain hominins. We can see on the bottom here, right? This time period of variability associated with the last appearance date of Australopithecus, this one of Australopithecus afarensis, the appearance of stone tools, older one here, and so on and so forth. Okay, and he originally connected, and I think he probably still writes about this, that these time periods of high variability cause and can explain these milestones of human evolution. For example, origin bipedality, encephalization, the evolution of a larger brain, right? These things that he hypothesized were associated with being a generalist, dealing with these climates that swung back and forth quite dramatically in short time periods. Now, what's interesting is that if you remember from the figure I showed several slides ago, right, that del O18 curve recording paleo temperature, we see that paleoclimate gets more arid and more variable through time, okay? And so these two hypotheses, specifically turnover pulse and variability selection, right, because we have time periods of when there is a shift towards aridity and a shift towards high climate variability. And both of them predicted that you get the speciation extinction of certain taxa during these time periods. These two hypotheses kind of make a lot of the same predictions, particularly when you should expect this cluster of speciation and extinctions. However, what Elizabeth Verba and Rick Potts did in their studies is they took the fossil record at face value, right? So again, that Elizabeth Verba figure I showed earlier where the temporal range truncates, right? The fossil lineage disappears from the fossil record. She treated that as the time of true extinction. But remember, from my previous lecture that the LAD, last appearance date, does not reflect the true extinction date, okay, particularly when the fossil record is incomplete, which it for sure is. Conversely, the first appearance date, the FAD, does not reflect the true speciation date, okay? So when you account for the incompleteness of the fossil record, which you have to do statistically, there is no evidence of these turnover pulses at the predicted time periods, okay? Whether you're going according to the Verba's turnover pulse hypothesis or Rick Potts's variability selection hypothesis. So here, this is a paper that was published recently. I was on this, and so I actually calculated these statistics. But going from about 3.5 million years ago to the present, okay, on the x-axis, on the y-axis, we have basically what is the speciation rate in blue, extinction rate in red, okay? These are the time periods that you would expect turnover pulses according to either the turnover pulse hypothesis or the variability selection hypothesis. We don't see either red or blue curve spiking at these time periods, okay? So it appears that that signal of a turnover pulse, as found by Verba and even Rick Potts, that was due to the incompleteness of the fossil record. Okay, so does climate change actually influence hominid evolution? Okay, maybe you thought that the answer was an obvious yes, but this is the most basic question you can ask, and this is actually kind of controversial. Okay, this was really spelled out in a paper by Kay Berensmeyer. You've now come across that name multiple times, published in 2006. Climate Change and Human Evolution. Okay, very simple title. Sub-question is, what can we learn about cause and effect relationships between climate and human evolution from the late Cenozoic? So because of Verba and Rick Potts' work, it was all the rage to study climate change and understand how it influenced human evolution. But Kay said, hold on, okay, because these are different spatial scales, okay? And I showed this figure earlier, so you already know, right? Global scale, that Del 18 curve, the continental scale, the dust record, right? Site scale, this is soil carbonate, Del C13, that will go over in a future slide. But basically, the takeaway here is that these organisms, they experience environments at the local site scale, right? You could think about just your everyday life for a given day, where you walk around, which buildings you enter, right? How much you walk, you're traveling like, I don't know, kind of miles in the single digits, maybe tens of miles at most. Maybe a little bit more if you're driving around, right? But you're not driving all across the US, okay? You're not driving all around the world, right? And so the way you interact with your environment is at these smaller scales. And so when we go from these local scales to larger and larger and larger scales, you're getting further and further removed from what these animals are actually experiencing on the ground, okay? And as I will show in a future slide, as you go to larger and larger scales, or if you have these scale differences between large scale and small scale, there's a lot of processes that happen in the middle that can alter the global climate signal, that can mute it, amplify it, okay, all these different things can happen. I'll show you a more specific example later on, but right, what you experience here in Colorado can be very different than like what a penguin experiences in Antarctica, okay? Right, I think penguins are in Antarctica. Okay, last section, reconstructing paleoenvironments. And so again, I treat paleoenvironments here as reconstructing these habitats at the smaller local site scale. Basically the spatial scale that hominins are actually interacting with their environments. Very much something like this ancient landscape that you would see here. Okay, so this section, we'll just finish out and I'll go over the different ways that these paleoecologists reconstruct ancient environments, aka paleoenvironments. So one straightforward way to do it is just to look at the fossil plants that are preserved. And this gives us an idea of what the vegetation was like back then, aka paleovegetation. So you go to a fossil site, you look for fossil plant remains, whether that's fossil pollen or fossil phytoliths, basically these kind of little rocks that plants make them kind of grittier to eat. It's a defense against like grasses, for example, have this, and it wears down the teeth of grazers more. So it's kind of a way to defend themselves from being overconsumed by grazers. But these fossil plant remains tell us about the abundance of these ancient plant taxa. Okay, so here's an example. These are all microscopic of a phytolith and pollen. Okay, what you get is if you can identify in the fossil phytolith or pollen whether they belong to trees, they perhaps even, I mean, this isn't what I study, but I imagine maybe even certain tree species, certain grass species, okay? And you can see how abundant these different fossils are through time. Okay, so here's an example from trees. And then this gives us an idea of how abundant trees are through time at a given site. Same with grasses, okay, herbs, and all these other things. Okay, so that's just a straightforward way to reconstruct what the vegetation was like back then. And the abundance of these fossils reflects, yeah, presumably how abundant these plants were on the landscape in the past. This isn't very common though, particularly at early hominin sites, because the type of sites that early hominins are found in, they typically don't preserve fossil plants very well. Okay, this one is a lot more common. Fossil mammal community structure. So much like how different types of plant fossils tell you about the types of plants on the landscape, different mammal taxa reflect different habitat vegetation due to their particular diets and locomotion, the way they move around, right? In woodlands, you particularly get colobus monkeys. They're very well adapted for living in trees, eating leaves, as we'll learn in a future lecture. So if you find a colobus monkey fossil, very likely there were trees there, right? If you find zebra fossils, that means there were grasses around. Zebras are adapted to eat grass, to run around in grass. Okay, if you find a fossil hippo, that indicates there must have been a large body of water nearby. Same with if you find fish fossils, crocodile fossils. And if you find things like impala, that does usually imply some type of mixed grass and woodland habitat. It's kind of more mosaic. Yes, and researchers have published on how common these different taxonomic groups are at their fossil site, which again presumably reflects how common these different vegetation types are at their site. This is kind of a more sophisticated way to do it, if you will, but this was a really seminal paper when it was published decades ago now, but by Andrews and Nesbitt Evans. I guess he's a lord. I don't know how you get that title. But the paper is titled, yeah, Patterns of Ecological Diversity in Fossil and Modern Mammalian Faunas. Okay, and what these two authors and researchers noticed is that here we're going to have 4 different habitat types. Okay, and for each one, there is a representative site. And this is A, B, C, D, E, F, G, H on the x-axis are different body size categories. On the y-axis is number of species. Okay, and as you go from A to H, you go from smaller body to larger body. So we see that in these kind of more wooded environments, you get a lot more small animals, fewer large animals. And as you move towards a more open environment, that kind of flattens out. Less small, more big. Okay. You also see differences across these 4 habitats. If you look at the way different species move around. Okay, so SMG or LMG is a large terrestrial mammal. SMG is small terrestrial mammal. A is arboreal, those who live and move around in trees. As you can imagine, if you find a lot of those, a lot of trees around. S, scansorial, things that climb trees and other substrates, AQ, aquatic, AAR, aerial, things that fly around, right? And basically the takeaway is that these different habitats have different distributions, okay? They have different number of species in each of these different categories, depending on which habitat you live in, okay? And you can do the same thing with diet. Okay, so these are different diet categories, and you see these distributions are different depending on which habitat you live in. And the idea is that you go into the fossil record. So this is a site, again, Olduvai Gorge. I didn't go over taxonomic order, but these three, right, size, locomotor adaptation, feeding adaptation, right? The idea is you can look at these distributions, and because we've shown in modern sites today that different habitats, different vegetation types have different distributions, right? That means there's this environmental signal in these different categories. And so you could kind of look at your fossil site and the distribution and try to match it up to what we see in the modern, okay, and see which modern habitat is best reflected by these distributions, and thus infer at Olduvai which of these vegetation distributions is best reflected by these fossils. That's kind of the logic here, and this is still done today. There are more sophisticated statistical techniques that can be used to assess this, but yeah, it has its strengths and weaknesses. One is it assumes the modern is the key to everything, and that the past would have been like the modern, when it could be that the past was fundamentally different for different reasons. One obvious one is that humans didn't live back then and alter habitats as much as they do today. Another reason is there were very different fossil species that lived in the past that really have no analog today. Okay, so how do they fit in all this? How might they have changed the ecology of the past compared to today? And I'll talk about a different reason that kind of makes comparing the modern and fossil a bit of an apples to oranges phenomenon. That's known as time averaging, which I'll talk about in a future slide. Okay, here's another way to use the mammals found at a hominin site, using those mammals to reconstruct these ancient environments. Here we see Kay Barentsmeyer again. This was her postdoc researcher. Okay, so already finished grad school, but not quite a professor yet. So studied with Kay Barentsmeyer at the Smithsonian. Now he's a pretty famous paleoecologist in his own right. But yeah, the title of this paper, The Expansion of Grassland Ecosystems in Africa in Relation to Mammalian Evolution and the Origin of the Genus Homo. Okay, so here's a figure from that paper, right? On the x-axis, we have time. Y-axis, we have grassland indicators. What does that mean? Basically, this is the relative abundance of grassland indicator taxa at a site. Okay, so if you're at a site, and let's say 50% of the fossils we find are like zebras, wildebeest, things that like grasslands, okay, then that would be, well, it doesn't even go that high, but yeah, 0.5. So that's what this is tracking. So the fact that this is increasing through time is showing that at this site, it is going from more closed woodland habitat to more open grassland habitat over time. And you could see, I forget exactly why this is shaded in gray, but these authors connect this habitat change to, for example, extinction of Australopithecus, the appearance of Paranthropus and Homo genera, and appearance of stone tools. Okay, mammal ecomorphology. This is another way we can use mammal fossils to reconstruct paleoenvironments. So an organism's morphology tells us about its diet and habitat, right? Just like Darwin's finches, just like the examples I gave earlier. But here is one well-known relationship that if you have like a softer diet, so to speak, a lot of leaves, fruits, you have shorter teeth. Okay, something like these two. And if you eat grass, okay, which again have those phytoliths, very abrasive, you have taller teeth, right? So it's like an arms race between grazers and grass. So grass, you try to evolve more phytoliths, so you wear down the grazer's teeth more. Grazers, to combat that wear, you evolve taller teeth. Okay, so even if you wear it down a bunch, which definitely happens, okay, you still have teeth, you can still eat. So basically these adaptations tell us something about its diet and therefore the habitat. So if we have animals that have very tall teeth, right, ate a lot of grass or grazers, that probably tells us there were a lot of grasses back then, right? Here's another example. This is an astragalus, aka an ankle bone of an antelope, and its shape tells us something about how it moves around in its habitat, okay? And what we see is that if you live in a woodland environment as an antelope, you have evolved an ankle shape that is more flexible. Basically, you can dart left and right very quickly, right? Which is good for escaping predators, dodging around trees. Whereas if you move around in open grassland, your ankle is more rigid. Okay, your ankle has not evolved to move left and right very sharply. Okay, in fact, it's quite the opposite. It's rigid. You don't have much lateral movement at all. Your adaptation is to go straight as fast as you can. Okay, you want to outrun your predator in an open grassland. There are no trees to dart around to try to lose your pursuer. Okay, and so I don't want to go into the details, but yeah, these different vegetation habitats, different ways of moving around, which is reflected in the morphology of these ankle bones. And you could take all these measurements and do a sophisticated statistical analysis. And what you can get is something like I'll show you on the next slide. Okay, paleoenvironments of the Shangura Formation, famous paleontological site, Paleocene Ethiopia, based on ecomorphology of the bovid astragalus. Again, astragalus being that ankle bone. And so these are geological members through time, basically different strata through time. And by analyzing and measuring a bunch of ankle bones of these antelopes in these different time periods, and through a statistical method, this researcher was able to calculate the proportion of these different habitat types in these different time periods. Okay, yeah, forest, heavy cover, so still a lot of trees, but not as much as a forest. Lighter cover, fewer trees. Open, very few trees, if any. And you can see how these proportions have changed through time. Okay, and again, ultimate goal is to reconstruct these pale environments to understand the selective pressures experienced by hominins. Okay, the effects of time averaging. So what is time averaging? This affects, in this case, I make a connection towards those paleoecologists that use the mammal fossil record to reconstruct pale environments. But I suppose this can really be applied to any paleoenvironmental reconstruction method. And so, actually, let's just keep it to mammals. So what we know about mammals when they die and leave their skeletons on the landscape, and how common and frequently burial occurs, is that the skeletal input rate outpaces burial rate. Okay, so what that means is animals die. This is K. Berensmeyer again. Their bones are left on the landscape. Okay, let's say 1 year, 1 season. This is an elephant. An elephant came along and died. Okay, its flesh has been eaten away by carnivores, bacteria, whatever. And let's say a year later, another individual, let's say wildebeest, comes along and dies. Okay, its bones are left on the landscape. Flesh is eaten by carnivores, bacteria. And the year after that, you get other individuals, so on and so forth, such that you start getting the skeletons and bones of individuals piling up, or at least found on the same location on the landscape, that probably never saw each other during their lifetimes. Okay, and again, that's because the rate at which these animals die and their bones are left on the landscape, that's much faster than the rate at which burial happens. Okay. And so what you get then is the mixing of non-contemporaneous fauna in fossil assemblages. Okay, here's a way to show this. Okay, ecotone is just a fancy name for a boundary between two habitats. So if we start at time 1, okay, we have this gray habitat. We have this black species, species X, that is adapted to the gray habitat. We have a white habitat with species Y. Okay, we see the habitat boundary shifts over time. So what was mostly gray is now mostly white, and thus has mostly species Y. And the next time period it shifts back again. Okay. And what happens is in a time average fossil assemblage, these 3 time periods all collapse on each other, right? Because all their bones are remaining on the landscape when they die. It's not like after time period 1, the bones of species X get cleaned away. No, they're still on the landscape when the habitat shifts to mostly white habitat with species Y. Okay, so then what happens is all these bones survive on the landscape and thus mix species X and species Y because this boundary has shifted over time. Okay, and then finally, maybe there's a burial event, the bones get buried, and these things all get buried together and may eventually become a fossil assemblage. But you get this mixing of species X and Y in this fossil assemblage, even though these species never coexisted during their lifetime. That's time averaging. And that is, and I alluded to this on that Andrews and Nesbitt-Evans slide, but that could be one reason in my mind, because I studied this phenomenon for my dissertation. So I believe this is like probably the explanation for why a fossil assemblage might not look anything like a modern mammal community. Because fossils, even a fossil assemblage that you find where these fossils are preserved together, those fossils can represent together hundreds of years, thousands of years, right? Whereas we studied modern living communities on the time scale of days, weeks, months, years, maybe decades at most. So this scale difference, time scale difference, is like apples and oranges comparison. Okay, I think this is the last paleoenvironmental reconstruction method I go over. This is something called stable carbon isotopes. Oh, I should also mention that the oxygen isotopes I referred to earlier, Delo-18, those are also stable, meaning they're not radioactive, they're not going to decay. Okay, they remain stable through time, their ratio remains stable through time. And so here we have a ratio of a heavier carbon isotope to the lighter, this is the most common, carbon-12 isotope. And this ratio is quantified using this del C13 measure. And how does this work? Okay, so this is a busy slide, but I'm going to walk you through it. So you have carbon in the atmosphere, okay, in the form of carbon dioxide. And that carbon dioxide has a certain ratio. It has a certain del C13 number, again, measured in parts per mil. In this case, is negative 6.5. And so plants, they uptake this carbon dioxide for photosynthesis, right? Now, would you know it, the photosynthetic pathway for trees is different from the photosynthetic pathway for grass. And so what that means is because these different photosynthetic pathways, they fractionate and they deal with carbon differently. And so when trees take in atmospheric carbon dioxide, they change the del C13 signal to become more negative, way more negative, negative 26, whereas grasses make it more negative, but not as much, negative 12. Okay, so already we see different del C13 signals, even though the atmospheric carbon signal is the same. Okay, it's because of the different photosynthetic pathways between trees and grass, aka C3 plants, C4 plants. Now, those animals, for example, giraffe that eats the leaves, the giraffe, right, takes in the carbon from the plants, incorporates those carbons into its tissues, including teeth. Now, again, as I mentioned in the last lecture, teeth, very common in the fossil record because they're pretty much already rocks and fossils to begin with. So this is good news. And then animals, if they eat grasses, right, they'll incorporate that carbon signal in the grasses into their teeth. Teeth become preserved, right, at a given fossil site. And so what that means is these animals, their teeth are recording how much grass and how much trees and leaves and shrubs they ate. Okay, so you can actually drill and geochemically analyze fossil teeth to see whether they ate more trees or more grass. Pretty cool, right? And so presumably you drill enough teeth, you start getting an idea of how abundant trees and grasses were on the landscape. The other thing is that, for example, trees, okay, they'll drop leaves. Those leaves will decay in the soil, become part of the soil. The roots of trees will also respire and exchange carbon with the soil. And so what that means is the soil carbon is equilibrating with the tree carbon signature, okay, such that these carbonates, these kind of rock formations, if you will, not quite rocks, but whatever, these formations that form in the soil, they record the overlying carbon signal from the trees. Same with those that form here, they're recording the overlying carbon signal from the grasses. Okay, and these soil carbonates, known as pedogenic carbonates, they actually also preserve in the geological record. So if you find them, you can, and they look like this, you can also geochemically analyze them to see whether the overlying vegetation was more trees or more grass. Okay, and so the takeaway here is that anytime you have stable carbon isotope analyses, the more positive the per mil signal that reflects grasses, aka C4 vegetation, whether you're analyzing fossil teeth or fossil soil carbonates. And the more negative the per mil signal, it's more of a trees and shrub signal, a more C3 signal. Okay, so yeah, this basically says what I said earlier. You can analyze geochemically these ancient soils, also known as paleosols, and fossil teeth to estimate proportion of grasses versus trees or shrubs on the landscape. Okay, so here's an example done using analyzing ancient soils. Okay, these are two different sites, green and red, but you can see here's time on the vertical axis. Okay, here is the del C13 signal, okay, which you can translate into percent woody canopy cover or these vegetation categories. So you can see within these sites how vegetation has changed through time. And as is commonly the case, it's gone from more wooded towards more grassy over time. Okay, yeah, you could also drill fossil teeth. As you can imagine, you could drill fossil mammals. You can also drill hominins themselves, and people have done that. So here we have time going from older to younger. Okay, we have different hominin taxa. You could see who they are. But what we see is that the earlier hominins, they had a more C3 diet, eating leaves from trees or shrubs or probably fruits. And then over time, it becomes more variable. So they're able to incorporate different types of vegetation types. And it also does get more C4, aka more grassy. And I should note, it's not only, particularly for hominins, it's not only the plants you eat, but also the animals you eat. So if you're eating a grazing animal, you're going to get more C4 signal. If you're eating a more browsing animal that eats leaves and fruits, you're going to get that more C3 signal. Okay, and yeah, this is the final slide before the conclusion slide. So I just want to note the importance of scale. So this is kind of emphasizing what I said about that Behrensmeyer 2006 paper. Global climate may influence local habitats, but local ecological conditions will always overprint that global signal. Okay, so here's an example of that. Okay, so this is these are 3 different sites that are quite close to each other, and so there's no scale here, but this is modern Lake Turkana. Okay, so on the left side is a site called Nachakui. On the north side is a site called Chungura. On the east side is a site called Kubifora. Okay, you can see those three are reflected here. And that indeed, so yeah, time is on the vertical axis. On the x-axis is the percentage or relative abundance of grassland indicators. So very much that Bobet and Berensmeyer method I talked about earlier. But you see in each site, it does trend towards grassy environments, which is a pattern now that I've mentioned multiple times, because you see that at many, many sites through time. But you see that they're different in terms of this Shangora is always towards the left, so it always has more trees. Kubi4 is in the middle, Nachakui is on the right. And why is that? Well, the differences in grassiness is due to the structure of this depositional basin and its water source. And so I'm not going to get into this because you need to know some background geology, but the way these basins form, okay, and the structure of this particular basin is what makes Nachakui quite dry, Shangura very wet. So Nachakui, a lot of grass, Shangura, a lot of trees, Kubi forest somewhere in the middle. So that's complete reflecting these more local geological conditions that's overprinting the global signal. Okay, so again, that's what challenges, as Kay Berensmeyer pointed out, using global climate signals to try to explain something that really is a local on-the-ground phenomena, specifically the evolution of these hominins. Okay, so in conclusion, hominid paleoecology is a study of how hominin taxa are distributed across time and space, and the ecological reasons for this. In paleoecology is a lot of paleoenvironmental reconstructions. In fact, paleoecology pretty much equals paleoenvironmental reconstructions, whether that's large-scale paleoclimate or more local-scale paleoenvironments. Okay, there are many ways by which we reconstruct these different large-scale paleoclimates and smaller-scale paleoenvironments, right? We covered, frankly, a bunch. And I presented the main paleoclimate hypotheses in human evolution, aka savanna hypothesis, turnover pulse hypothesis, variability selection hypothesis. But how true they are is still really an open question. So a lot of work that needs to be done in paleoanthropology.